Hello everyone, we are the Neurophysiology Laboratory from the University of Balearic Islands, and our work focuses on studying how the nervous system functions by analyzing biomarkers, behavior, and processes such as oxidative receptors.
In our lab, we work with different animal models, specifically rats of the Spragueii and Wistar strains, which are widely used in neuroscience research due to their reliability and well-established characteristics.
To address our objectives, we employ a combination of complementary methodologies. First, we conduct behavioral tests such as the Barnes maze and the rotor, which allow us to evaluate cognitive and motor functions. Subsequently, we perform biochemical studies, including the analysis of oxidative stress through the use of specific biomarkers. Currently, one of our main ongoing projects aims to investigate long-term neuroinflammation. To achieve this, we analyze different populations of glial cells, such as astrocytes and microglia, assessing how their alterations are related to neuronal function and neuronal survival.
The selection of the appropriate experimental method depends on the specific research question and the level of analysis required. Choosing the right method is essential to ensure that the data obtained are both valid and meaningful, and that the conclusions accurately reflect the biological processes under study.
In addition to in vivo models, we also incorporate new approach methodologies, or NAMs, which aims to reduce reliance on animal experimentation.
In our lab, the primary NAMs we employ are cell culture systems, including both primary cultures and established cell lines. In particular, we use co-culture models, such as neuron astrocyte and neuron microglia systems, which allow us to study cellular interactions in a controlled environment.
These approaches are especially valuable for complementing our in vivo studies, as they enable us to investigate specific molecular and cellular mechanisms with greater precision. Moreover, they contribute significantly to reducing the number of animals used in research.
These combined strategies align with the principle of the 3 Rs: reduction, replacement, and refinement, as well as with the 4R, the responsibility, which emphasize the ethical obligation of the researchers to ensure the animal welfare and the scientific rigor. By integrating NAMs with animal research, we aim to maximize the scientific validity while we minimize the ethical impact.
NAMs play a complementary role in our research, as cell culture systems allow us to study specific cellular and molecular mechanisms under highly controlled conditions with great precision and reproducibility. However, they cannot fully replicate the complexity of living organisms, where multiple systems interact dynamically over time, limiting the study of processes such as integrate neuronal function, behavior, and long-term systemic effects. Therefore, combining NAMs with in vivo approaches enables us to leverage the strength of both systems and achieve more robust and comprehensive scientific outcomes.
What both the general public and policymakers should better understand is that the use of animal models is not arbitrary, it's justified and carefully regulated.
At the same time, it's important to recognize that methods such as NAMs are not replacement, but complementary tools. They allow us to reduce the animal use and gain deep cell mechanistic insights, but they cannot fully reproduce the complexity of a living organism.
Continued support for both ethical regulation and the development of innovative alternatives is essential to advance science. Thank you for your attention and for supporting responsible and evidence-based scientific research.